Let us talk about bacterial growth in this video lecture. Bacterial growth in culture media. Not in the nature. Bacteria grow in size as well as in numbers. When they increase in size, a time comes when they divide into two cells like this through a process called binary fission. So most bacteria undergo this mode of cell division or reproduction or multiplication. But there are other methods like fragmentation, conidia formation, and budding, etc. But here we are talking about binary fission because most of the bacteria, they follow this method of reproduction. So a bacteria divides to form two similar cells after a certain time period, and then again these two cells will divide in the same time period into two daughter cells again, and the process will go on, and the number will double up at certain period of time. This time period is called the generation time. So this is the first generation, this is second generation, this is third generation, this is fourth generation, and so on and so forth. In each generation, the number doubles. So from one cell, if you start from one cell after the first generation, there will be 2 cells. In the second generation, there will be 4 cells. In the third generation, there will be 8 cells. In the fourth, 16 generations. And like this, it will double up in every generation. So we can write this also in this manner: 2 to the power 0, 2 to the power 1, 2 to the power 2, 2 to the power 3, 2 to the power 4, and like this. If there are n number of generations, then it will be 2 to the power n. The final number of cells will be 2 to the power n. So we will see the mathematical expression of this in maybe our next class or next video lecture. So the bacterial cell increases in this way, and this occurs in a kind of a culture that is called batch culture. There are different types of culture techniques, like the batch culture, continuous culture, or the synchronous culture. But the growth pattern that is being studied in most literatures are in the batch culture. A batch culture is a kind of culture which occurs in a closed vessel, like in a conical flask. When we study the bacterial growth, we usually take some amount of media in conical flask. We plug it with non-absorbent cotton and sterilize it. After that, we inoculate it with a certain bacteria, and then we measure the growth of the bacteria in this particular closed vessel without further adding any nutrition or taking out any nutrition. Only samples will be taken out from here in test tubes or in incubates that can be measured. The optical density of this will be measured with the help of an instrument called turbidimeter. Even colorimeter does the same job. There is another instrument called nephalometer that measures the reflected light, not the absorbed light. So this is called a batch culture. A batch culture may also occur in fermentation or industrial microbiology, where large vessels are used. In that also, batch to batch fermentation processes are there where the media is added once and for all, and that is being inoculated with a certain with a particular strain of bacteria. The bacteria grows up, it utilizes the substrate present there, converts it into product, and finally when the process is completed, the product is taken, the spent culture is taken out, and it is subjected to downstream processing for the production of the particular secondary metabolite. So this is how a batch culture operates. In a continuous culture, there will be a provision of addition of fresh media when the fermentation is going on, and at the same time there will be a provision of taking out the spent culture from the vessel, so that the process always remains in a steady state. The cell number always remains in a particular constant state when the production, when the yield is highest. It is first studied at which particular stage the yield is highest. At that particular stage, the cells are maintained in a continuous culture. But in a batch culture, all the different phases of bacterial growth can be observed. So we have seen there are 4 distinct. If you plot the number of cells with respect to time, so this is time in hours, say, and this is cell number. Then you will say that the growth pattern of the bacterium growth goes like this. So there are 4 phases of growth: 1, 2, 3, and 4. These phases are known as lag phase, number 2, log phase, log or exponential phase, number 3, stationary phase, and number 4, death phase. All these 4 phases can only be observed in a batch culture, not in a continuous culture or in a synchronous culture. There are between these stages there are some transition stages, like when the cell shifts from one stage to the other, there are transition stages also. These are the transition stages, the corners. During these phases, what is happening? The cell is shifting from lag phase to log phase. So this phase is known as phase of accelerated growth. This one is the phase of accelerated growth. The second transition phase that occurs between the log and the stationary phase is the phase of decelerated growth. Phase of deceleration, when the rate of growth decreases. Again, the third transition stage is this one, when the live cell number decreases very fast, and that is also accelerated. This phase is called accelerated death phase. So there are 3 sub-phases or transition phases in addition to these 4 distinct phases of growth. Why these phases occur? What is lag phase is all about? Lag phase is the first time when the cell is transferred from one medium to the other, when it has been the new inoculum has been given into this vessel. What happens if the previous media was not the same? Then the cell will need some time to adapt to the particular environment, a particular nutrient present in the media. To break it down for its nutritional requirement, the cell might require switching on some genes, some operons, and that's why it takes some time for that, and that is why the lag phase is all about. As soon as the process of breakdown or catabolism starts, the cell enters into the next phase, that is called the log phase. When the cell number doubles every particular after a particular time period, that time period is called generation time. At every generation time, the cell doubles. That is also called doubling time. So that phase is called the log phase. It occurs from here to here when the growth rate is constant, And then a time comes when the culture, the nutrient in the media, it is getting exhausted, and the cells are deprived of media now, but little amount of media is still left. That is why the cells are still growing up, and as many cells are produced, as many cells die, and that's why the total number becomes constant like this. It is parallel to the baseline. But after some time, all the nutrients present in the media are exhausted, and in fact, certain toxic materials are being added up in the media. These toxic materials are inhibitory for the growth of the culture, and in fact, the bacterial cells start dying very fast due to this increase in the toxicity, change in the pH, etc. And also due to the anaerobic environment that is increased when the time goes on. So the cell go into the next phase, that is called death phase. So these are the reasons why these phases exist in a batch culture, and I think this will be good enough for your preparations in the undergraduate level. Thanks for your attention.